Hello and welcome to the web series, ZeroPM Pieces. In the ZeroPM Pieces, the researchers in the project will tell you about one piece of the ZeroPM puzzle that they're working on. So without further ado, here comes a ZeroPM piece.
Okay, hello everyone. My name is Baygildiev and I'm a postdoctoral researcher at Vrije Universiteit Amsterdam. And today I would like to tell you about the methods and approach that we use to identify PMT and other emerging substances with effect-directed analysis in water samples from the Upper Rhine River. And of course, this work was not performed only by myself, but also with a group of authors that, and I'm very grateful for them. And you can see all of them on the slide. Of course, it's also not a secret that water is contaminated with thousands of chemicals, and actually it's more like a chemical cocktail. It's contaminated by different sources like wastewater treatment plants, like industry, like agriculture, and many other sources. And the two questions that most often we want to know answers to which chemicals, as public and as scientists, of course, which chemicals contaminate water, and especially PM substances. So I'm not the first one who is making presentation in the ZeroPM project. So you all know these notorious substances. And the second question, of course, that these chemicals pose a hazard, and there are two possible ways to address the questions and two approaches. That is suspect and non-target screening based on LC-HRMS and bioassays. And we combine those two approaches in the method that is called effect-directed analysis. Here you can see the workflow or experimental setup that we applied in our research for the identification of bioactive compounds. And it consists, of course, of water samples that are going to be analyzed, all the pre-concentration step. In our case, it was solid phase extraction, because still water, of course, is contaminated, but the level of contaminants are still not extremely high, so samples should be significantly pre-concentrated. Then the obtained extract is analyzed by the reversed phase HPLC in our case, and it's actually analyzed two times. First portion goes to the high resolution mass spectrometer or HRMS, and another portion goes to the fractionation device, Fractionate, with the help of Fractionate, eluent is spotted into the bioassay plates. And then based on the response that is can be detected in the bioassays and the data that we obtain using HPLC-MS, and we can identify these bioactive substances. Further, I will split this apart, this scheme, and go one by one. So first, I would like to start with the solid phase extraction, as we were looking for the PM compounds and other emerging compounds, and we all know that PM compounds are rather polar, so we need to have really broad chemical space, and using just of only one sorbent can significantly restrict the chemical space. That's why in our research we applied so-called multi-layered SPE. So this cartridge that we applied, it consists of a mixture of 5 different sorbents, the main goal of which to pre-concentrate and to sorb as many compounds as possible. And with this combination, it can sorb polar compounds, hydrophobic compounds, and ionizable compounds like cationic and anionic. Then the extract from the SPE is analyzed, as I said previously, within LC-HRMS. We applied a column that is capable of that can offer us increased retention of polar compounds. but for the rest, it was rather commonly used mobile phases like water methanol. And the gradient also was optimized for the increased retention for the polar compounds. Detection was performed using the quadrupole time of flight mass spectrometer, and we did that in two modes, in positive, of course, and negative modes. So, and every extract was analyzed 4 times in full scan mode and in DDA, as that stands for data dependent acquisition mode, which is a crucial step, of course, for the identification of chemicals, because it allows us to get a mass spectrum. We also applied our in-house QA/QC mixtures and University of Athens retention time indexes mixture to be able to predict retention times for unknown features. Then the next part of the scheme is fractionation, and that was performed with the Fractionate device that was developed here at the FU. Using the Fractionate, effluent from the column can be spotted into the bioassay plates. In our case, we used 2 plates, and then plates are evaporated and analyzed in the corresponding assays. And based on the result that is observed in the assay and based on the data that you see at the bottom, LCHRMS data, we can identify or try to identify chemicals that pop up in the active fractions. Next step is the assays that we used. And first assay is transferrin binding assay or TTR binding assay. The core element of this assay is a TTR, transporter protein, and this is primary carrier of the thyroid hormone, thyroxine, or T4. And in the presence of competitive xenobiotics, T4 can be displaced from thyroxine, so from TTR, so less thyroxine, less T4 is transported, and that can presumably lead to the thyroid hormone system disruption, so, which is not very good, of course. So, and the method is the bioassay is based on the displacement of the T4 conjugate with feed C that is fluorescent. So that, as you can see above on the slide, and if this conjugate binds to TTR, there is a high fluorescence signal, and in the presence of competitive binders, part of this conjugate is displaced from TTR, and so this leads to the decrease in the fluorescence. So if decrease in fluorescence is detected, that means that and this for this fraction, this decrease in fluorescence is significantly different from the background. This means that some competitive binders probably could be present in these fractions. Next assay, second assay that we applied is Alevibrio fishery bioluminescent bioassay, or AFB bioassay. And it utilizes the bioluminescent bacteria, Alevibrio fishery, that when it is healthy, it emits light, and in the presence of some toxic compounds for that bacteria, it's the luminescence is decreased because the well-being of the bacteria is also diminished. For example, here it is a positive control for that. We applied triclosan. So again, if the inhibition or decrease in luminescence is observed, that means that possibly some active compounds, some contaminants are present in this fraction. For the data processing, so important thing, of course, of the identification of chemicals is the data processing step, and we perform that with the vendor software, broker data analysis, and broker metaboscape, and annotations were performed hierarchically. First with libraries of it's like Mass Bank of Europe and Mass Bank of North America, and then with a suspect list like KEX screen and KEX screen with a simulated metabolites. And also we specifically developed two suspect lists in this study. That is a list of TTR binding chemicals and Alevibrio fishery bioassay active chemicals that we performed by screening of the literature data and by our internal in-house experience. And this is significantly facilitated and streamlined identification of chemicals that might be in charge for the bioassay response that is detected in the assay. Annotations were assessed with a total annotation quality code. For this, we used different parameters like mass deviation, retention time, isotopic pattern, MSMS score, presence of MSMS spectra, and the intensity of the feature that is higher than intensity of the blank. And this code was translated to the Szymanski confidence levels. So the lower is the level, Szymanski level, the higher confidence of identification is. So here on the slide, you can see all the possible combinations of the tag code that lead to the Szymanski confidence levels. And of course, the highest level of identification is level 1, and it can be achieved only if reference standard is available. But level 1 of chemical is not enough to claim, even if it was detected in the active fraction that is in charge of the response that is observed. The next step of confirmation is also the testing of the level 1 identified chemical in the corresponding bioassay. And if this chemical has concentration dependent assay in the corresponding assay, then this chemical can be not only a level 1 chemical, but level 1 bioactive chemical. And further, it's really nice if it can also be semi-quantified or quantified for the further evaluation of the contribution of this chemical to the response in the fraction that was observed in the bioassay. In our case, we did it with the standard addition method, and the formula and the equation for that is presented on the slide. In that case, if with the contribution, with the known IC50 or IC20 of this chemical in the bioassay, and within its semi-quantive or quantive concentration, contribution to the fraction response can be calculated, and we can know if there is a need for further examination of the data. And with this, I want to finish my presentation and quickly summarize that in our research, we applied sample preparation that were capable to pre-concentrate a wide range of PM compounds. We applied TTR binding and assay and Alevibrio fishery bioassay to the unfractionated and fractionated extracts of all 10 tested samples to reveal chemicals that may pose risk to human health and environment. And PM substances and other contaminants were prioritized for identification using those two assays and subsequently identified through suspect and non-target screening. And the data, we also created a database and suspect list of compounds that are active in those assays that help to streamline and facilitate identification processes. And for further details, I hope you will see them soon, because we haven't yet published that, and going to submit the manuscript soon. So for further details, yeah, I hope you can in future see it in the published manuscript. Thank you. With this, I want to finish my presentation, and I'm ready for questions.
ZeroPM, Zero Pollution of Persistent and Mobile Substances. This project has received funding from the European Union's Horizon 2020 Research and Innovation Programme under Grant Agreement number 101036756.